this is of course a staff round, but it's a very unusual staff round. And traditionally we start staff rounds with a clinical case. And we do that because the point of these rounds is to emphasise advances in medical science as they apply to the clinic. So this is about medicine meeting science in a clinical context. And today we're not going to have a case, and that's very unusual. And I had to think to myself as to how to justify that. And I mainly have to justify it to Patrick Sissons, who for many years was the stalwart supporter of this staff round and who was one of the key people insisting on the fact that we retain our immediacy to clinical medicine, and something I support. The reason I think we can buck with tradition today is because there are two reasons. The first is that this is a very important occasion. That the Harveian oration, which Steve is going to give for the second time here, is the most prestigious lecture supported by the College of Physicians. It's been running since 1656, apart from a few years off to rebuild for the Great Fire of London. They've fortunately broken with the tradition of everybody having to talk about William Harvey, which must have got a bit difficult after a couple of hundred years. And only 4 people from Cambridge this century have given the Harveian oration. That's Boris, Keith Peters and John Gurdon, and Steve is the 4th. So one reason for breaking with tradition is the fact that this is such an important event. The second reason is that Steve actually epitomises the interface between clinical medicine and science, and everything he's done really speaks to what the staff round usually does, and for that reason we really need no excuses. So I'm going to tell you briefly about Steve, very briefly Steve, don't worry. So because everybody of course knows Steve, but he trained at UCD in Dublin, spent time in Oxford and at Harvard before ending up in Cambridge. I met him actually 32 years ago in Oxford when even he was just a registrar. Everybody said, that guy Steve O'Reilly, he's going places. He hadn't done anything then, done no research, nothing. And they were right, yeah. When Oxford people are going places, they generally come to Cambridge. So Steve is, of course, a fellow of the Academy of Medical Sciences and the Royal Society, and a foreign member of the National Academy of Sciences of the USA, and has a knighthood and all that stuff, and lots of prizes. I'm going to tell you no more about him, but hand over. Thank you, Steve.
Thanks, Ken, for that very generous and over-generous introduction. I think you were meant to tell people that this might go on for a little bit more than the hour, because the Royal College of Physicians gives you a generous hour for your presentations. And if any of you need to leave, I won't take offense. So it's highly appropriate that I'm talking today at a grand round, because it was as a young doctor in the early 1980s when the concept of the grand round was at its heyday, and at the Hammersmith it was a particularly wonderful place to be, and the grand rounds were tremendously dramatic events where nervous young junior doctors would work for weeks to work up particularly interesting cases, would have a scholarly discussion of the science behind it. Sometimes science from the very institution itself will be presented alongside the case, and then that will be followed by a battle royal of the gathered grandees of medicine at the Hammersmith. And this was something that I think William Harvey would have really appreciated, because he was a great protagonist of both clinical medicine and science, and he exhorted through this lecture fellows and members of the college to seek and study out the secrets of nature by way of experiment. I've enjoyed reading about him in preparing for this lecture, and found out what a gentle soul he was. One of the nice quotations from his Lumleian lecture notes, that he handwritten notes, was he reminded himself not to praise or dispraise other anatomists, for all did well, and there was some excuse even for those who were in error. And that was, I think, a rather gentle sentiment, one we might remember in these days. However, the Hammersmith in those days wasn't always like that, and in fact, sometimes the atmosphere was considerably more grand guignol than grand round. So I am actually, Ken, going to start this grand round with a case, and I'll start with a patient M, who's a 43-year-old Caucasian woman who's single, who works in a supermarket and was referred to diabetes clinic because of persistent poor glycemic control. And when I saw her in clinic, she said to me cheerily, "My insulin isn't working again." And the story is as follows: that she was markedly obese since she was a child, and type 2 diabetes was diagnosed at the very young age of 27. when she presented with weight loss and vaginal thrush. But her main thing at that time was how delighted she was at how much weight she'd lost as a result of the presentation with diabetes. And then she went on multiple oral medications, insulin from the age of 33. Of course, the insulin continued to increase in dose, her weight piled on. And she eventually, after a couple of visits, admitted that over the years she'd had frequent periods of insulin omission as the only way she could find of getting rid of her lifelong excess of weight. Her medication was large amounts of insulin in divided doses, but only very intermittently, taking a variety of other cardioprotective and anti-diabetic medications. She, on examination, as you see, was obese, but markedly centrally obese, and she had been more obese. She was hypertensive, she had the beginnings of retinopathy, she had the beginnings of sensory neuropathy, she had the beginnings of nephropathy with microalbuminuria, and she had very poor glycemic control. So this is not at all an uncommon scenario. We see it all the time in our type 2 diabetes clinics. And what are the questions that are raised by her? The questions, I think, are as follows. The important ones are some of them. Why has she been persistently obese from such a young age? Why has she developed type 2 diabetes so early in life? And what can we do to improve her health, well-being, and prognosis? And that generalizes to the questions that I'm really going to try and address today in the talk. Why are some people particularly susceptible to the adverse metabolic effects of nutrition? Why are some people particularly susceptible to the obesogenic environment? And what therapeutic opportunities do we have in obesity-related metabolic disease? So obesity, counter to a lot of what you may read these days, is not, in fact, a new disease. The Paleolithic sculptor of the Venus of Willendorf some 25,000 years ago couldn't possibly have dreamed up the anatomy of this particularly obese woman without having experienced that visually. Hippocrates thought quite a lot about obesity and recommended that obese people should eat once a day, take no baths, and sleep on a hard bed and walk naked as long as possible. So we've gone very far really since those days. The 18th century jailkeeper in Leicester, Daniel Lambert, was very obese, but there were many obese people in that era, and in fact in the Georgian era, Thomas Short commented that no age had seen more instances of corpulency than our own. However, it is true that obesity has become much more common. Data from the United States Center for Disease Control showed a particular marked inflection point starting around the 1980s and perhaps plateauing off a bit now in adult obesity, particularly the transition between people who were overweight becoming obese. But that trend had been going on for over 100 years. This part of the division is an 1890 Union Army Caucasian Eastern Coast of American volunteers, several thousand of them, and they at that age had about 3% of them had a body mass index over 30, and you just flick on 100 years to exactly the same Northeast American demographic, the same age, male sex, and the increase in obesity prevalence is tenfold over 100 years. So there has been a marked increase over a century in the prevalence of obese individuals, and of course that's not restricted to the United States. This is pretty universal, and we of course are in the nasty dark red bit now, where we have among the highest prevalences of obesity in the world. And we have to explain what the reasons for the secular increases in prevalence are. It's not my particular expertise. I'm fortunate to share a building and share discussions with many superb population scientists. But briefly, I think that the secular increases are probably largely explicable on simple grounds, on the fact that it has never been cheaper, more readily available, more attractive and less satiating to get calories than it has been in the last 20 or 30 years. We have marked changes in the food environment. We also live in an era where we're expending less calories in physical activity, both at the home and work, and less thermogenic demand because of heating and cooling than we ever had before in our history. And to me, these represent a sufficient set of reasons why we've had a marked increase in the prevalence of obesity. Of course, there are many other suggested factors, such as antibiotics and environmental pollutants, your antenatal environment, your mother making you fat, social networks, your friends making you fat, and there are, of course, other genetic pieces of information out there which are actually well documented and probably do play a small role is that assortative mating, fat people tend to marry fat people, and differential realized fertility, where obese women produce more offspring than non-obese women. And this is pretty secure in most westernized countries. So they probably play a small role in the increase in prevalence of obesity in the last 50 years. But of course, why isn't everybody fat is a key question. So there's two sets of possibilities of why isn't everyone fat. We all have the same biology, and lean people make conscious choices to keep them lean, or they tend to be in environments that promote leanness. That's one very reasonable set of hypotheses you could put up. Or you might want to suggest that people have different underlying susceptibilities to being lean versus obese, and that could be impervious to environmental factors or could interact with environmental factors. And you won't be surprised that what I'm going to try and tell you in this talk is the following too, is that people do have different underlying susceptibilities to being lean versus obese, and it does indeed interact with environmental factors. And we know about the fact that obesity is a heritable condition because it's a highly heritable trait. Studies from identical twins, adoptees, and even identical twins reared apart give a very high heritability estimate. And the most remarkable study is that of Stunkard, published in the 1990s, who went to Sweden, collected information on children who had been adopted early in life, but identical twin pairs who'd been separated and brought up in different families. So a true separation of the gene and the physical environment. And his conclusion was remarkable, that genetic influences are substantial, that childhood environment has little or no influence. There was almost no correlation between an adult's body mass index and the siblings with which they'd been brought up, but a very powerful and strong correlation with the identical twin that they'd never seen. And the visual manifestation of this in a separate piece of work, this is not the Swedish twins, but they're just to give you a visual cue. Here are some sisters born on the same day, sharing their same environment, sharing their same intrauterine, or very similar intrauterine and same postnatal environment. They're sisters, but they share 50% of their genes, and they come in all shapes and sizes like you and your brothers and sisters do. But if you look at sisters born on the same day who share 100% of their genes, it'd be hard to look at that and say, I don't think that genetics has anything to do with obesity, BMI, or adipose distribution. So obesity is very simple, and we could all go home after this slide because it is actually energy stored equals energy in minus energy out. But of course, we could conceivably store energy in different ways. We could store it as fat tissue, and we could store it as non-fat tissue. I often dream that I could go out for a big curry one night and put all the calories into a vigorous beard growth that I could cut off in the, cut off in the morning, but unfortunately, adipose tissue is developed to be the sort of tissue that is beautifully professionally designed to store energy. And we have to do a remarkable job because throughout our lives we consume vast amounts of calories, and yet we don't all oscillate between Luciano Pavarotti and Kate Moss throughout our lives. We tend to stay along relatively stable trajectories. And the homeostatic control of mammalian energy stores is something that has been studied for over 70 years, and workers both in the UK, Gordon Kennedy and Adolf, were the first to show that if you take a rat, which normally continues to grow all its life, and you calorically restrict it for a couple of weeks, and then you put it back in a free food environment, it will eat back until it reaches the trajectory that it was once on, suggesting that it has a way of sensing its nutrient stores, sensing what it's lost, and it wants to go back to that original trajectory. And where would that be sensed? That was the question that they first started answering, and it was human observations that first suggested where that should be sensed. It was people like German physicians like Fröhlich who had shown that people who were normal weight during life and then they suddenly presented with headaches, hyperphagia, and became progressively obese and had tumours in the hypothalamus. And many of those were reported in the 19th century, and it was first suggested that the hypothalamus was the centre of controlling of body weight energy through those natural human experiments. That led to a more scientific approach to that in the 1940s and 50s, when Hetherington and colleagues electrolytically lesioned parts of the hypothalamus. And if you lesion the medial hypothalamus, you get a massively obese animal that overeats. And if you lesion the two lateral hypothalami, you can create an animal that starves itself to death, that simply won't eat, with tiny sub-millimeter lesions in the lateral hypothalamus. The first, I think, critical experiment in the modern obesity era was done not but 2 miles from here in the old Addenbrooke's Hospital, which is now the Judge Institute, by a chap called Harvey, who worked in a small MRC unit. And he did a beautiful experiment, one we couldn't do these days because we wouldn't be allowed to do parabiosis in this country, or it would be very difficult. Parabiosis is the Siamese twinning of newborn mice so that they part share circulation, so that factors like hormones can cross the circulation, but not major bulk transfer of nutrients. And so what he did was parabiose 2 rats, then created an electrolytic lesion in one that became enormously obese, and then he demonstrated remarkably that the other animal, the parabiosed animal, lost interest in food, didn't eat, and became very skinny. And it wasn't because he was being carried around on the back of the big fat guy. It was because if you lesioned that animal, it also overate and became so it's perfectly capable of getting to the food, but it just lost interest in eating. And that was the first suggestion, that there is a circulating substance which is present in mammals, which provides information about the state of nutrient stores in the organism. And that then moved forward 20 years to the Jackson Labs in Bar Harbor, Maine, where collections of of mice with abnormal phenotypes were being studied. And the OB mouse, a recessive obesity phenotype, was parabiosed again, this time by Doug Coleman, to a normal mouse. And remarkably, when you parabiose the OB to the OB, it cured the obesity of the OB mouse. So this looked like this OB mouse was lacking in a circulating factor, which was present in the normal but not in the OB mouse. And it took the power of modern molecular genetics and about 7 or 8 years of tough molecular cloning in the very difficult era for molecular genetics for Jeff Friedman to finally hone down and find the leptin gene and report it in 1994. And this was a remarkable discovery. An adipocyte-specific hormone was going from the adipose tissue to the brain and informing largely through receptors in the hypothalamus and elsewhere in the brain to decrease food intake, increase thermogenesis, and increase physical activity. And this has ushered in a fantastic era in obesity research, where, as well as the long-term signals related to the long-term signaling of leptin, there are short-term signals and satiety signals post-meal. And those signals help integrate our short-term satiety when we want to finish a meal. And in recent work that Fiona Gribble, Frank Ryman, myself, Tony Coll, and Johannes Gross from Takeda did, we've actually added another hormone to that set of hormones, the second orexigenic gastrointestinal peptide, insulin-like 5. And these discoveries have started to come into obesity management. Liraglutide is now licensed in the United States and is the largest selling anti-obesity medicine. Liraglutide is a modified form of GLP-1, one of the products of these enteroendocrine cells, and it is now licensed for management of obesity in the US. My own part in this little story started when I came here to Addenbrooke's in 1991, having done a postdoc in Harvard. And my idea really was that it was the power that we needed to exploit of extreme human phenotypes. And that was partly inspired by the story of the discovery of the LDL and the whole cholesterol homeostasis system by work of Brown and Goldstein. And around the same time, Rick Lifton, and I had heard him speak when I was in Boston, was applying the same sorts of approaches to trying to sort out the mess of hypertension by going to extreme subtypes of hypertension to try and understand what was going on in there, and discovering that primary disorders of renal sodium handling were dominant. And I wanted to apply that to metabolic phenotypes, particularly the insulin resistance and the metabolic syndrome. And I was keen to try and look out for opportunities to exploit these sorts of approaches. Remember, at the time, PCR was only coming into the general lab around late 1990s. It was very early days of molecular genetics, way before the revolution. So the idea of targeted genetic analysis was just coming in, and candidate gene analysis was not as trivial as it now is. And my first foray into the area of obesity came through this patient who actually came to my regular clinic when I was still a senior registrar at Addenbrooke's hospital, and she presented with reactive hypoglycemia. We found her to be relatively hypoadrenal, and she had hypogonadotropic hypogonadism. But she also had lifelong obesity, but particularly severe in childhood, even requiring admission to hospital as a child. And I could tell long stories about this, but to cut a long story short, we worked out that she was not processing a whole range of prohormones, that including pro-ACTA, pro-melanocortin, pro-insulin, particularly causing her reactive hypoglycemia. And joining me at the time was the senior registrar, Robert Jackson, in the lab. And Robert was a fantastically diligent and hardworking and dedicated young guy. And he decided he would take on the difficult job of actually cloning the one candidate gene we thought might be abnormal, which was prohormone convertase 1, which was not cloned in the human genome. So we had to clone it and identify it and then see if we could find the mutations. And we did, and we discovered that this patient had a compound heterozygosity for 2 mutations in the PC1 gene. We then, of course, got very interested, and particularly when Nature published this paper in December 1994 with Jeff Friedman. And in 95, referred to me was by Dr. Shella Mohammed, a clinical geneticist in the John Radcliffe Hospital, were these 2 children. They were severely obese from an early age, hyperphagic, very much like the human phenotype of OB mice. And early on, back in 1995, we decided before, again, the leptin gene was not cloned, but we thought we'd have a good idea and we'd take a skin biopsy and see if we could get a tiny bit of fat that was hanging off the skin and see if we could get cDNA from these children. And indeed, we succeeded to get a tiny bit of cDNA, and the full-length leptin was present in their skull. So they weren't lacking the leptin gene. We then went on, and that's Carl Montague, John Prinz, and I, to look at sequencing. And we had to clone, PCR sequencing of fragments was very difficult at that time, so we had to actually clone the cDNA and actually put it into clones and sequence clones. And the day we cloned that, the controls didn't work. So we just had to compare it against published sequence. And we got terribly excited because down in here, and Carl told me, well, it's really exciting, there's something different from the published sequence. But of course, what you see in this lab book here is so the control sequence was not correct. And indeed, it looked like this. So there was not an insertion deletion, and we got rather downhearted and went away. And thought about, we did a lot of other things that year, and there were a lot of other exciting things going on. So we thought we'd park that because there was no obvious other candidate gene we could look for at the time. The story moves on to when Sadaf Faruqi joined the lab as a welcome clinical training fellow to undertake a PhD under my supervision. And we thought as the first thing we'd give her to do was measure leptin in a range of samples, but also in these 2 children. And she came back and said, I can't find any. And I said, well, you're very new in the lab, so you must be wrong, so do it again. And she did it again, and she said, I still can't find any. So we went back to the sequencing gel and looked more carefully, and to our horror, but also delight, is we noted that of these 5 Gs in a row, there should actually be 6. And we'd missed the fact that there was a homozygous deletion of a G in a set of Gs, and this child had a frame shift mutation found by this sexy new sequencing technology, capillary, found very easily in the new form of sequencing, and causing a mutant leptin product that was not secreted from adipocytes. So that then led to our paper on the first evidence that humans could be deficient in leptin, and that was associated with the same phenotype as in the mice. And the discoveries of those genes really was a bit of a milestone, because actually obesity had not really been taken seriously as a biomedical problem by and large, and only a few small number of labs and adepts were working on them. And in a very short space of time, we discovered the first 2 genes that had caused humans to be obese. So that caused a lot of stir at the time, and of course, most importantly, it pointed to a therapy. And Sadaf and I went on and managed these patients and gave them recombinant leptin, and with remarkable effects that have occurred and been reproduced many times since. We went on to find more highly penetrant obesity genes, and that is Sadaf convinced me. Initially, I wondered whether we just had 2 pieces of really good luck, and she said, there's got to be more out there. And she definitely convinced me, and with her drive and passion and commitment, to increase the number of samples we had from the small number in the lab to a very large cohort. And the creation of the Goose cohort is now the manifestation of that commitment to make this going from a small arena of activity to a major one. But we needed high-quality laboratory sciences to work with us, and we were very lucky at the time. Giles Yeo had finished his PhD at Sidney Brenner's lab and brought all his molecular biological expertise to the lab. And somewhat later, Tony Cull joined us, and bravely, for someone who was joining as a fourth-year clinical registrar, actually threw himself into the generation and phenotyping of mouse models, which is something we had no expertise on, which we desperately needed in certain features. So he's done a magnificent job from going from ground zero to being able to generate and phenotype highly complex animal models. And it was a very exciting era. We and others around the world discovered lots of interesting genes. Probably the most important is the melanocortin-4 receptor gene. This is present, mutations in this are present in probably 1 in 1,000 people in the UK. It's probably commoner than Duchenne muscular dystrophy or cystic fibrosis as a medical condition. And so that was really a very important, I think, discovery. But probably philosophically, the most interesting thing is all these genes, when we found them, were being highly expressed in the brain, particularly in the hypothalamus. And that led us, and Sadaf in the clinical research facility did a magnificent job phenotyping and beautifully documenting the detailed clinical phenotype of these patients, showing that not only was leptin deficiency associated with hyperphagia, but also individuals with melanocortin-4 receptor mutations were obese largely because they ate too much. And we could even calculate how much they would eat by looking at how much cyclic AMP they could generate from their mutant receptor. And that correlated directly with the amount of food intake eaten at a test meal. We also learned an enormous amount about human biology, reproductive biology, immunology, and indeed the central control of appetitive by studying patients before and after leptin repletion, including in collaboration with our neuroscience colleague Paul Fletcher and Ed Bullmore, the fantastic effect of leptin on the visual response of the brain to food-related stimuli. Giving these children leptin totally transformed. Showing them food-related pictures in the unleptin-treated state, there are areas of the addiction and reward in the brain lit up like beacons in response to, like someone with cocaine addiction being shown the paraphernalia of cocaine addiction. But as soon as leptin was given, just within 2 or 3 days, before any weight was lost, this change in blood flow in these areas in response to food stimulation was totally reversed. So here we had this simple hormone circulating in our blood and yet having a profound effect on what we would think is higher level functions in our brain. We continue to discover obesity genes, and Sadaf is now leading and has led for some time the discovery efforts in obesity, and that's continued to be exciting and highly productive. And we've been very fortunate with the collaboration with the Sanger head of principal investigator Inez Barroso, who's been a wonderful collaborator in many of our genetic studies over the years. But the people who are skeptic about our work still say, well, that's all very well in rare children, but how about common obesity? Surely this mechanisms can't be going on in common obesity. And colleagues in the MRC epidemiology unit and elsewhere around the world collaborated on these massive genome-wide studies. And what we're looking at here is where the most likely gene next to the SNP that associates with obesity, where is it expressed in the body? Where is the dominant story here? Is it digestive? Is it endocrine? Where are those genes being expressed? And in fact, it's remarkable. The genetic associations that influence body mass index across this room are almost entirely central nervous system dominant expressed. And in fact, many of these genes overlap with the monogenic disorders that we have previously described. And can we finally link that common gene expression in the brain with some sort of behavior? Well, our sadly a deceased colleague who just died very tragically early last year, Jane Wardle, did beautiful work in childhood obesity. She developed a childhood eating questionnaire that reliably predicted the risk of children becoming obese in association with food responsiveness and satiety responsiveness. And layer on genetics to that, we can see that actually the same genes that influence BMI, this is the number of alleles that you have for these risk genes, the same ones that determine your BMI also associate with diminished satiety responsiveness. So finally, we have, I think, a workable model of how genes and environment might interact to produce obesity. Obviously, during conditions of famine, nobody gets obese. It's pretty simple. When food is limited, only those with high genetic susceptibility become obese. But of course, now we're in a situation of abundance, and even those of average genetic susceptibility are becoming at risk, but the very high ones are very prone to becoming obese in the current environment. The one thing that in this field we struggle with in the way that people who study, for example, hypertension don't struggle, people don't go around pointing fingers at people saying, yo, look at that person with a high blood pressure, is social discrimination. And it's very easy to find if you just peruse the average newspaper every day, every week, just casual and comments. But of course, it's a bit more shocking when you actually find it coming from members of one's own profession. I'll just give you an example of a certain Dr. Bob Berry, thankfully now retired, I believe, a radiologist. Here's what he wrote on his blog in 2010. I'll just leave you read that for a second and just reflect if that was said about any other social group, ethnic group, any other person with any other condition, would this person not only be struck off the medical register, but also possibly be prosecuted? So just worth thinking about. Because this does have serious impacts on people with obesity. A beautiful study done by Will Dietz a few years ago showed that if you match young people for exactly academic attainment, family status, family income, and you match for BMI, BMI is a strong predictor of poor social and poor economic outcome by the age of 30, largely, as he thought, about because of discrimination against individuals who are obese. And that's because, in general, often considered to be lazy and feckless. But of course, they're not all lazy and feckless. Here's William Taft, who was the 27th only person to be president of the USA and Chief Justice of the Supreme Court during their lifetime. He's struggled manfully with his weight all his life. His diaries are wonderful. I recommend you reading them. They're a fantastic documentation of what a terrible struggle it is and what a difficult it is to try and lose weight when you're genetically predisposed to gaining it, although we don't know his precise genotype, of course, as yet. But he was seriously overweight all his life. But I'll read a little piece to you from the diary of his secretary, Ira Taft. He was the chief of males, actually, in the White House. And he wrote in his diary, "The president dieted all right, but not when he could escape supervision. I remember once when I accompanied him on a journey to Ohio. When we got on the train, leaving the doctor and Mrs. Taft behind, the president began to perk up. He also apparently began to think about food, although it was 10 o'clock in the evening. The president appeared at the door of his sitting room. I took one look and knew what was on his mind. 'Anyone seen the conductor?' he asked. The conductor came running. 'The dining car,' Mr. Taft began shyly. 'Could we get a snack?' The conductor looked surprised. 'Why, Mr. President, there isn't a dining car on this train.' The president's suntanned face turned pink, with perhaps a few splashes of purple. 'Norton,' he called in a cold voice. 'Mr. Norton,' Norton was his private secretary, 'there is no diner on this train.' Norton reminded Mr. Taft that they'd had dinner at the White House and assured him they would not go without breakfast. He recalled that the president's doctor had warned him about eating between meals. The president brushed him aside, turning back to the conductor. 'Where's the next stop, damn it?' he asked. 'The next stop where there's a diner.' The conductor believed it would be Harrisburg. Mr. Taft glared at Norton and addressed the conductor and said, 'I am President of the United States, and I want a diner attached to this train at Harrisburg. I want it well stocked with food, including filet mignon.' He silenced the secretary's protest with a roar. 'What's the use of being president,' he demanded, 'if you can't have a train with a diner on it?' So not all obese people are lazy and feckless, but they certainly seem to be food motivated. So what about the adverse consequences of chronic overnutrition? Really what we're talking about is what happens when our energy intake exceeds our energy expenditure. Of course, we term it obesity, but obesity is rather a trivial term, simply a descriptive term for an expanded mass of weight of triglyceride within adipose tissue. What we're concerned about is its adverse effects. Its adverse effects on mechanical gravitational adverse effects, and cancers and metabolic disease. And clearly the obesity mass itself is what has that effect on those mechanical and gravitational, on our knees, on intra-abdominal pressure with reflux esophagitis, and our pharynx with sleep apnea. But it's not entirely obvious why such expanded mass of triglyceride would lead to cancer or indeed metabolic disease. And perhaps there are some more subtle pathways from energy imbalance to those disorders, which we might be able to explore. The key intermediary, at least a key intermediary between overnutrition and metabolic disturbance, is the association of obesity with hyperinsulinemia or insulin resistance. And what we mean by insulin resistance is people who need more insulin in their blood for an equivalent amount of glucose lowering. We have a very glucocentric view of insulin resistance. So these people all had, in this early study, all had normal fasting glucose, but as you got heavier people, they needed more insulin for the same degree of fasting glucose. So by definition, they were insulin resistant. So insulin resistance became a phenotype of great interest, and one that really started in my lab here. I started in my lab in 1991 working on, and with tremendous colleagues, initially Anna Crook and later Robert Semple, David Savage, and more recently, of course, with Inez Barroso again. We looked around the country and around the world, indeed, for people with extreme insulin resistance and discovered a range of individuals with loss of function mutations and some people with markedly enhanced insulin action with the gain of function mutations. And a whole range of these disorders were found over the last 20 or 25 years. And I'm going to briefly jump from all of that fundamental basic molecular discovery to something else that we learned by simply not just studying these in cells, but by studying the patients in vivo and looking at their whole organism metabolism. And we discovered actually that when you have disordered insulin signaling, yes, you do share some phenotypes. You share hyperinsulinemia and you share an increased diabetes risk. But in typical metabolic syndrome, you have high triglycerides, you have low HDL, you have low serum adiponectin, you have high liver fat. And people who have insulin signaling defects don't have any of those things. In fact, they seem to be almost protected from many of those things. So quite simply, message is insulin resistance as we know it as common or garden insulin resistance doesn't look like what happens when insulin signaling pathways don't work properly. It looks a bit different. One condition, though, we started studying many years ago, which does start looking much more like metabolic syndrome, are individuals who can't make fat cells or can't make enough fat cells or can't make enough lipid within fat cells, and they have so-called lipodystrophies. And far from being healthy and lean, these people have accelerated metabolic syndrome. They develop all of the features of the metabolic syndrome at a very, very early age. And over the years, particularly in more recent years with David Savage and again also with Inez, we've discovered or co-discovered and worked with others to discover many of the genes causing lipodystrophy. And I'm just going to focus on two here, which I think, not because they're common, but because they're particularly illustrative of mechanism. And people with heterozygous perilipin 1 mutations have dominantly inherited metabolic syndrome. They get all the features of metabolic syndrome. They have limited fat cell expansion. You can see this mother and two daughters have got very masculine fat distribution. And these individuals have a C-terminal mutation which causes a frame shift and a nonsense a jumbled sequence to be put at the C-terminus. They express both the mutant and the abnormal form of perilipin. But what is this perilipin? Well, it is only expressed, perilipin 1, on the surface of the unilocular fat droplet within the white fat cell. It truly is a fat cell specific protein. And it sits here on the surface of this enormous lipid droplet, which looks terribly boring, but in fact is a beautifully organized organelle, which we couldn't really live healthily without. And here is what perilipin is doing in health and in the fed state. Perilipin is doing nothing, because actually it's not interacting with any of the two breakdown enzymes. The first enzyme that breaks down triglyceride, the second enzyme breaks down. It's simply sitting with its partner CGI58, while the business of triglyceride synthesis goes on in the fed state. But in the fasted state, I mean this not over prolonged fasting, even overnight, your resting metabolism, particularly your cardiac metabolism, is very heavily dependent on free fatty acid. And that free fatty acid has to come from somewhere. And a large amount of that comes from your adipocytes overnight. And what happens there is that phosphorylation events occur on perilipin and on hormone-sensitive lipase, which do the following things. The phosphorylation event on the C-terminus knocks CGI58 off perilipin and causes it to interact and bring the first initiating enzyme of lipid breakdown to the surface of the triglyceride droplet and starts producing free fatty acids and diglyceride. The N-terminal phosphorylation event brings the second enzyme, hormone-sensitive lipase, to the surface of the droplet and then again produces, and you get this beautiful coordinated cascade of triglyceride breakdown and free fatty acid release. And to cut a long story short, after much work and beautiful work by a very talented postdoc, Sheetal Gandrotra, who's now running her own lab in Delhi, we have fantastic a model of what's happening here. And what's happening in this condition is that essentially CGI58 is unable to bind ever to the of the mutant form. Remember, there's only one mutant form, the other is wild type, so this is enough. And you get a chronic activation of early lipolysis, irrespective of nutritional state. So even in the state of being fed, these fat cells are still releasing free fatty acid. And that aberrant release of free fatty acid is sufficient to result in all of the features of the metabolic syndrome that we're interested in, every one. The second example I'm going to give you is side C, and again, this is a real example of rarity because there's one human being in the world who we know to be lacking in this protein side C, which is necessary for the formation of single droplets from multiple small droplets. It's a biophysical action on the surfaces of the droplet. And if you don't have side C, you've got multiple tiny fat white droplets and much more active lipolysis within that cell because of the sheer surface area of the droplets. And we discovered that in a human some years ago, and David Savage, and this is work that I was not involved in, entirely driven by David and his collaborators in Korea and Hong Kong, did beautiful work to cross this mutation into the OB-OB background and show, for example, that even in an OB-OB mouse, you get tiny amounts of fat. But despite the animal becoming leaner, you get hugely fatty liver, large amounts of triglyceride in the liver, and profound insulin resistance. If you give insulin to a normal animal, you have to infuse loads of glucose to compensate. If you infuse insulin into this animal, you need almost no glucose. So despite being leaner, it's severely insulin resistant. And what's intriguing, because there is so much emphasis on adipose inflammation as the cause of insulin resistance. In fact, in this case, David showed that in fact, you cure the adipose inflammation. You take away the crown-like structures. You have a pristine-looking adipose tissue. You reduce the inflammatory markers, both in the cells and in the serum. And despite that curing inflammation, you worsen insulin resistance. A complete dissociation between adipose inflammation and insulin resistance. These elegant results are, I think, relevant to a broader question. And that is, how do we get from obesity to insulin resistance in muscle and liver? It's not intuitively obvious why a large expanded fat mass leads to defective insulin action in liver and in muscle. So how do we get from sustained positive energy balance to these defective forms of glucose handling? I think if you asked a large number of diabetologists and diabetes researchers around the globe, you'd find that the commonest hypothesis currently is that the expanded fat mass leads to a range of adipose inflammation, including the production of adipocytokines and the reduction of adiponectin, and it's those adipocytokines that are causing the defective insulin action and defective glucose handling by liver and muscle. But I think if you look back over a longer period of diabetes research, you can come up with an alternative hypothesis, and that is that the fat mass is insufficiently able to expand to cope with the chronic excess of energy balance. And when it reaches the limits of its safe adipocyte storage, you start to get deposition of ectopic fat in other places in the body. And it's that ectopic fat that is wreaking the havoc by mechanisms we still have to understand in liver and muscle. To ask that question in a general population, we recently worked with our colleagues in the MRC epidemiology unit and Inez Barroso at the Sanger Institute to ask a question of what are the common genetic determinants of insulin resistance and the metabolic syndrome in the broader population. And to do that, we studied nearly 200,000 people, and we came up with a score of 53 variants in the genome which consistently associate with high insulin, high triglycerides, and low HDL cholesterol. Now, the colors here are reflective of how we selected these traits, so they're not surprisingly, they're significantly increased and decreased respectively for insulin, HDL cholesterol, and triglycerides. And reassuringly, in separate populations, these SNPs associate with diabetes and coronary artery disease. Where the interesting thing starts to come in is the relationship with body fat and body mass index, where you can see a lot of blue here. And to take this further, if we start looking at the relationship between these SNPs and fat mass within the legs and buttocks, you find that these SNPs associate with not increased but actually decreased fat mass. We took this forward into a population of patients with an extreme form of abnormal fat distribution, so-called partial lipodystrophy type 1. And these individuals have got markedly reduced fat on the legs and markedly increased central adiposity. And we found that they were highly enriched in those insulin resistance variants in that syndrome. And we went back to the sort of DPIC, the bioinformatic analysis that I showed you before in relation to obesity. And this time, instead of the brain, which if you'll recall, was the predominant tissue in which variants causing obesity were expressed, when we looked at these genes that were causing the insulin resistance, we found that in fact, the key tissue was abdominal and subcutaneous fat and adipose tissue, and here adipocytes. So this is a problem primarily of adipocytes. And taking this work forward into cell biology, by knocking down these genes and showing what they did to cells, we were able to demonstrate that these SNPs were affecting genes which had a primary impact on adipocyte differentiation. So I think it'd be useful to give a sort of overview of this idea through a rather simple analogy, what I've called the sodden or soggy bathroom carpet model of metabolic disease. If you think of normal metabolic health as a situation where you have a normal sized bath, where you have a dynamic between energy intake and energy expenditure, and you reach a certain level of stored triglyceride in your bath of adipose storage capacity, that's metabolic health. Our conventional view of metabolic disease is that we have increased energy intake over and above our expenditure, and we fill up our bath, and that spills out, if you like, on to the carpet, and that's the metabolic adverse effects, the insulin resistance and diabetes. But perhaps what our data is suggesting that we should think about it in a slightly different way. Perhaps, for example, people have different sized baths, so that some individuals with only very minimal excess of intake over expenditure already start to have their wet carpet at much lower levels of imbalance between intake and expenditure. And this, I think, is an interesting concept, and one that might indeed even explain why certain ethnic groups, South Asians for example, are particularly prone to the adverse complications of overnutrition at what we would consider relatively modest levels of body mass index. So to sum up and go back to a slide I showed you towards the beginning, what have these genetic studies taught us about how to avoid type 2 diabetes? They've certainly, I think, told us that the brain is hugely important in determining whether we do or don't maintain energy balance. I think in particular they've told us that surprisingly the adipose tissue itself, long thought of as a passive recipient of energy imbalance, is actually hugely important in either determining whether the fat is stored safely or unsafely. We have found surprisingly less genetic influences over insulin action in liver and muscle, although that may in part reflect the technical difficulties of measuring that in large populations. And in work I haven't shown you because it's come from colleagues around the world and the country, including Mark McCarthy in Oxford, Andrew Hattersley and others, the pancreatic beta cell and its health is a principal determinant of whether you transition from insulin resistance to type 2 diabetes. An organ system that's rather remarkable by its silence in all of this genetic work is in fact the innate immune system and indeed host defense. We see very little genetic signal implicating those organs in the etiology of these conditions, which contrasts rather markedly with a very large body of murine experimentation, which puts the immune system center stage. And I think this is something that we really need to reconcile. So back to our patient that started this whole story today. She sadly has had a triple misfortune. She's inherited alleles which drive her to acquire and retain calories. She's inherited alleles which impair her ability to safely store excess energy as fat. And sadly, she has lived in an era during which caloric exposure has been almost constant. So it's no wonder that she has run into such tremendous trouble with early onset obesity and early onset diabetes. And how can we, in practical terms, help her? Well, our best route, I think, is by what seemed to some a rather brutal approach of bariatric surgery. For those of you who are unfamiliar with bariatric surgery, many of you may think that it works by obstructing the intake of food or by causing malabsorption of food, but it does neither. It actually works through causing a change in the gastrointestinal rewiring and hormonal signals, such that after a small meal, one is much more satisfied and satiated than one was before having the surgery. And the data is accumulating markedly as to the benefits of this in patients such as patient M. Here, for example, is a recent study of a 3-year outcome of intensive medical treatment versus bariatric surgery for people with diabetes. Here, for example, here is the effect on their body weight, which is obviously dramatic in these two types of surgery versus medical therapy. Here's the effect on glucose control, with the glycated hemoglobin dropping markedly. And here's the effect on the number of medications taken by the patients, dramatically reducing the number of medications. Indeed, more than half the patients had no diabetes on no medical therapy. And that, of course, has great implications for cost effectiveness. And a recent review by the London School of Economics has concluded that the bariatric surgery is cost effective even in patients who currently are not recommended by our National Institute for Health and Clinical Excellence, and is more cost effective in diabetes mellitus than it is in regular obesity. So here, for example, is a patient that's recently attended our National Insulin Resistance Service, and I'm grateful to Anna Steers, who's the lead clinician in that service, for providing me with this data. This patient was, like our patient M, struggled with obesity and high body mass index, very poor glycemic control, very high glucoses, and multiple medications and multiple attempts to lose weight. And you can see that she is one of those people with low fat mass in the legs and limbs, but large fat mass centrally. And even though her body mass index was less than 35 and therefore not formally approved for surgery under the current regime, a special case was made, and in December 2012 she had bariatric surgery. And you can see the remarkable results here with marked reduction in weight, almost normalization of glycemic control, and complete cessation of anti-diabetic medication. A tremendous impact on this person's life and well-being. So in conclusion, I'd like to draw really 3 threads together. I think that what we've shown today is that extreme human phenotypes can continue to be hugely informative about biology and medicine, and that reflects previous talks and Harvey narrations of people like Archibald Garrod, David Weatherall, and Keith Peters. I think we need to emphasize that despite the fact we'd always prefer to work with humans, there is a continuing need for doing animal research in medicine. Endocrine and metabolic disorders affect interconnected physiological systems, and we simply cannot undertake the sort of detailed physiology in cells or in a dish. We do need a multidimensional strategy to tackle our overnutrition related disease, much as we've done, for example, with hypertension, with a successful combination of public health measures and the development of safe pharmacology. We need to change our environment, and this needs population health measures, and the main barrier for that is political inertia and the political benefits of maintaining the current status to many vested interests. But we do also need to continue to focus on the individual, and there will be people who continue to need therapy for their metabolic diseases. And they'll need better drugs, better devices, better behavioral therapies. And the main barrier to that is scientific ignorance. And what we need is scientific research, and what we need is scientific research done by the best brains in the world, wherever they come from. I'd like to thank my colleagues and collaborators, and I've mentioned them throughout this talk, so if I've forgotten to mention any particular individual, there have been so many that have contributed that I apologize to you now. Thanks to our referring physicians, our funders, but particularly the patients, participants, and families without whom this research program would not have been possible. So thank you for your attention.